Hi, nice to meet you all. I'm Jennia Michaeli, REI, Mount Sinai Fertility, University of Toronto, and I want to thank the committee and the organizing for this amazing conference and the opportunity to present today. I will mention that Madison Erb is an incredibly talented clinical embryology master's student, and we have done this project hand in hand, so regretfully she couldn't be here today, but she's kind of an integral part of this project. So today I'll tell you about how we explored the effect of fragment size, on follicular survival after slow freezing and thawing cortical ovarian tissue in the bovine model. So I'll briefly take you through the scientific rationale in the introduction, then we'll walk through our materials and methods, I'll present our preliminary results, and together we can discuss our next steps in this ongoing project. So I almost feel like I don't need any introductions for ovarian tissue cryopreservation in this audience, but I'll just mention that as more and more children grow into adulthood and surviving the battle with cancer, they face these long-term effects of chemotherapy, and luckily today we have something to offer. So ovarian tissue was declared non-experimental by the ASRM in 2019, and it was kind of endorsed by many of the fertility and the oncology societies. And in Canada as well, ovarian tissue cryopreservation is considered non-experimental and supported by the Canadian Fertility and Andrology Society. So despite being a very well-established practice, practiced globally very successfully with reports of live births from all over the world, it's a very multi-step procedure that has a lot of controversies along the way, and we actually heard in this conference the debate between slow freezing and vertification. So this project focused on a very small but crucial step of dissecting and processing the tissue. So in this quite recent and very elegant meta-analysis by Diaz and colleagues in 2022, they interestingly outlined how there is such a great variation in fragment shape and size from all the institutions in the world. So fragments can vary in shape. They can be strips, squares, or just variable size fragments. And the size can vary from as small as 0.5 millimeters to as large as over 160 millimeters in different institutions. So the aim of this project was to look specifically and to investigate whether the size of the ovarian fragments that is frozen through slow freezing and then thawing, and using the neutral red viability staining in the bovine model, whether it affects follicular survival of primordial follicles. For the purpose of this study, we divided the follicular sizes into 3 groups. We termed the small fragments small, and they measured 2 by 2 millimeters. Medium fragments were 5 by 5 millimeters, and the large fragments were 10 by 5 millimeters big. In terms of kind of the biologic rationale behind this is that we all think that the fragment size can affect the freezing stage by the efficiency of the permeation of cryoprotective media inside the fragments, and thus affecting the survival of the follicles upon freezing and thawing. And further on, when we transplant the tissue, fragment size might also have an effect on how quick the tissue becomes revascularized. So this was a very basic but interesting question to answer. So I'll kind of walk you through how we did it. I'm sorry, how do I go back? Okay, so we used the bovine model. So what you see here is a bovine ovary. We dissected it and slowly resected the medulla in a very traditional way, very similar to what is done in the Kyle Orvig lab, and until we reached the stage of a very thin cortical strip tissue, like a flat cortical cortex that was about 1 millimeter thick. Then we very precisely dissected the tissue in the 3 size groups. You can see an example here on the screen. Of course, we had multiple fragments of each size of 2 by 2, 5 by 5, and 5 by 10. Then we incubated the different fragments in cryoprotective solution and froze them with the slow freezing method, stored them for various period of time. And then we, on a different occasion, we thawed the tissue through transferring them in decreasing, sequential decreasing levels of cryoprotectant solution. And then, in a very important step, we separated the tissue on pre-labeled dishes that we really wanted to keep the size of the original fragments. These were our parent fragments because we had to redissect the tissue further into what we called microfragments in order to allow for the neutral red viability staining to penetrate the tissue. So we incubated the microfragments with collagenase and followed by neutral red viability staining for about 90 minutes. This is how it looked. These microfragments became very soft, and then we placed each microfragment, of course, always recording the original size of the fragment it came from, the original size of the parent fragment on slides. We covered them with a cover slip and gently pressed to create what is termed in the literature a squash preparation. These squash preparations create a very thin layer that enables us to look at these slides on the light microscope. I'll show you how the images looked in a minute, but I will mention that the challenge of counting on all the viable follicles was that it was a very live stain, and it kind of moved in front of your eyes. So what we did, we captured images, and then later on, we used a freely and online accessible tool that's called .goose in order to count all the viable follicles that we saw in these slides. So these are examples of some of the slides we had. These are two different magnifications. As you can see, in the bigger magnification, we can actually see the small primordial oocytes surrounded by primordial follicles with the oocytes surrounded by a thin layer of granulosa cells. And on the smaller magnification, you can see these small red dots, all representing viable primordial follicles. As you can see here, one of the challenges was that we could not identify the non-viable follicles from the surrounding non-stained stromal tissue. So this is a very early and ongoing experiment, so I'm presenting our preliminary results. As you can see here, we analyzed over 100 images, and we counted over 10,000 individual primordial follicles, and then we calculated the average number of follicles we saw in a field of view. So a field of view was actually the images that we took, and they measured about 1 millimeter by 1.5 millimeter in size. And our preliminary result, we found that there was less viable follicles in the medium-sized group, but I suspect that this is a statistical glitch. As you can see, we had more slides, we had more images in the medium-sized group. So currently, we're analyzing more and more parent fragments, so we can clearly distinct if this was kind of a difference that resulted from the natural variation in follicular density, and kind of answer to get a full answer to our questions whether the size of the fragments made a difference on follicular survival. So for us, these preliminary results kind of validated our slow freezing and thawing techniques, and we found that neutral red is a truly valuable method to identify viable follicles. Yet, we could not quantify the total number of follicles because we only see the living follicles. We cannot see the non-living follicles. And this is why, in our future directions and the next experiments we're going to do, we're going to counter stain with different stains to see the non-viable follicles, so we can have a total number of follicle count, and then out of that, we could calculate the proportion of the living follicles. And it's mentioned there to compare fresh and frozen thawed tissue. So we did conduct the experiments in fresh tissue and then in frozen thawed tissue, but it wasn't from the same ovary, from the same sample. So what we're going to do next, we're going to do both fresh and frozen thawed from the same sample, so we can compare the total number of follicles in the fresh tissue to the number of follicles in the frozen thawed tissue and see the proportion of viable follicles among onc follicles. And of course, eventually, we will continue the experiments. We plan to transplant the tissue into a xenotransplantation so we can assess the effect of fragment size on the step of revascularization and of the tissue once it's transplanted back to the patient. I would like to acknowledge all the help that we had in this project, and I'm happy to take questions.
Thank you, Dr. Michaeli. Looks like we have a question from Dr. LaRonda.
Hi, very nice work. We had set up that meta-analysis paper that you showed my student Ashley Diaz to do some of these experiments, which is exactly what we wanted the next steps to be. So I'm so glad that someone's taking this on. And I really want to applaud and support some of the future directions that you said because of some of the limitations in the current way that you had done your study. So if we, for example, consider that chopping up the tissue or like at various sizes might affect primordial follicle activation, which I think was one of our hypotheses, but also the cryoprotectant penetration and things like that. Would digesting that in order to see the neutral red viability stain, in an even longer condition, also do the same thing, and then just kind of normalize all of your follicle counts was one of my thoughts when you presented the data. But it looks like you're going to correct some of this in the future directions.
Yes, this is really, it's kind of, we're beginning this long-term experiment. These were our first, we just validated the technique because before we started, we didn't have all the equipment. We kind of validated that we can actually freeze tissue, we can thaw it, it's viable, we can use the neutral red stain, and then these are our preliminary results, but we definitely have a very well outlined next steps, and I'm hopeful to present it in the future with better answers to all of your good questions.
Yeah, I'm really looking forward to the H&E stain, so you can fix it right at the
yes.
That
so
point.
One of the pictures you saw, we did an H&E stain. So these H&E stains,
but before, without
exactly.
digesting it, right?
Exactly.
Okay.
It wasn't done at the right timing, just because we were just starting. But we will do it longitudinally with every step. We will do the H&E, and we can compare the viability. Exactly.
Okay. Great. Thanks.
Thank you.
Our next question.
Very nice work. I saw you were evaluating total follicle counts and viable eggs compared to different size fragments. But what about the percent viability?
Yes, so this is what I mentioned. We were hoping that we can differentiate between the viable and with the neutral red, because some people previously reported they could have identified viable follicles from non-viable follicles with the neutral red staining. We can only see the viable follicles. We couldn't differentiate the non-viable follicles in these specimens. This is why we would like to do H&E staining and counter staining. There are other stains that can stain the non-viable follicles. So we will stain both the viable and the non-viable, so we can see the total follicular count. Because, for example, in slides that we didn't see viable follicles, we couldn't tell, was it because there were no follicles because of the natural variability, the follicular density sometimes is variable, or because there were follicles that were not viable.
Okay, sounds good.
One last question. Thank you.
This might be the approach to make the standardized protocol for the tissue cryopreservation, and simply guessing that if you freeze a large pieces of tissues, you may need more the large volume of the equilibration and the vitrification solutions. And do you have any standardized the volume of the both media based on the size of tissues?
Yes, thank you for this thought. We kind of wanted to address more of the basic questions about, as I mentioned before, clearly all the different practices work well all around the world, so I'm not, it's not to say that there is someone who does it wrong. Everyone do it right. It was curious to us to know which way preserves more follicles, and every step kind of break this very long multi-step procedure and to explore a very specific question.
Thank you, Dr. Michaeli.